The carbon cycle is the process by which atoms of carbon are circulated within the Earth's biosphere. Carbon moves between the atmosphere, plants and microbes on and under the planet's surface, and in the oceans. Prior to the industrial age, the flux of carbon between these natural reservoirs was balanced. However, in the last several hundred years, humankind has been tipping the balance of the Earth's natural carbon cycle. The burning of fossil fuels for energy generation is a main contributor to this imbalance. Each year, human activities release approximately 6 billion tons of carbon into the atmosphere, and this number is rising. The carbon cycle is a closed system, and the accumulation of carbon compounds in the atmosphere is contributing to global climate change. Through the Carbon Cycle 2.0 initiative, researchers from the Berkeley Lab and the Department of Energy Joint Genome Institute are learning from nature to develop strategies that will enable us to produce enough energy for our needs while restoring the natural balance of carbon on the planet.
Laboratory. Now there are two laboratories that have his name appended to it, and the DOE Joint Genome Institute is part of both of those laboratories. Our workforce derives from both of those. It was Lawrence who's acknowledged to be the father of team science, that is bringing together physicists with biologists, mathematicians, and engineers to tackle the big questions of science that confront us. So we're going to start small tonight. And first with a little background about JGI. Got to have a mission, and our mission, after we sequenced the human genome, because 12 years ago we were formed to tackle about 11% or 3 chromosomes of the human genome, and in 2004 we were the first institute to complete our portion of the human genome and publish that analysis in the journal Nature, and then stepped back and looked at the rest of the biosphere. There are a lot of organisms under the sun, and many of which have to do with the Department of Energy's mission. We're going to hear about those this evening. So simply stated, our mission as a user facility, and that is catering to mostly the academic community worldwide, is to sequence genomes of plants, fungi, microbes, and communities of microbes, and make that information publicly available. So the fancy word for communities of microbes is metagenomes, and you're going to hear a couple of examples about that this evening. So our sweet spot are those targets within the umbrella of global carbon cycling, the area of bioenergy, and the other biogeochemical processes that are both moderated and mitigated by microbes out there. So first, what's a genome? That always helps. Got a genome? Plants have genomes. This is a small spike moss that was published just this last month by the Joint Genome Institute. It's got a genome. Trees have genomes, as do fungi, as do the moss below the fungi, as do the soil microbes below the roots of the trees themselves. Complex organisms, like this frog that was published in the journal Science last year by the Joint Genome Institute, has genomes, and even slightly more complex organisms have genomes as well. One can contend whether or not they are human, because while this organism has some 10 trillion cells in her makeup, with a genome in each cell, she has 100 trillion microbial cells in her body. So fundamentally, at a 10-to-1 ratio of microbes to Gaga, she's basically just a vehicle for microbes. So let's talk about genomes. The language of your genome, your total genetic complement, is reduced to just 4 letters of the alphabet, shorthanded here as A, T, C, and G, in long strings, long molecules arranged in this elegant double helix, packed into every cell in the GAGA body. When we say sequence a genome, what we really mean is to establish the order of those letters or nucleotides, because embedded in those long chains of letters is the functionality in elements that we know as genes and the flanking regions that have regulatory influence as well. So let's define carbon cycle. That is a carbon cycle. It's a carbon fiber bicycle. That's not what we're going to be talking about today. What we are going to be talking about is a more complicated system that we were going to hear about tonight. Microbes and plants and fungi have a lot to do with it. And what we did is put together a short video that we're going to swap over to now with the help of some friends, Frank Lehman, Lisa Cook, and Jason King, a recent graduate of the Expression College for Digital Arts. And in a minute and a half, we're going to try to capture your imagination with our version of the carbon cycle. So good luck. It's always dicey business, trying to swap files in the middle of a talk.
The carbon cycle is the process by which atoms of carbon are circulated within the Earth's biosphere. Carbon moves between the atmosphere, plants and microbes on and under the planet's surface, and in the oceans. Prior to the industrial age, the flux of carbon between these natural reservoirs was balanced. However, in the last several hundred years, humankind has been tipping the balance of the Earth's natural carbon cycle. The burning of fossil fuels for energy generation is a main contributor to this imbalance. Each year, human activities release approximately 6 billion tons of carbon into the atmosphere, and this number is rising. The carbon cycle is a closed system, and the accumulation of carbon compounds in the atmosphere is contributing to global climate change. Through the Carbon Cycle 2.0 initiative, researchers from the Berkeley Lab and the Department of Energy Joint Genome Institute are learning from nature to develop strategies that will enable us to produce enough energy for our needs while restoring the natural balance of carbon on the planet.
World premiere, and you're the first to see it. Thank you very much. So in the context of the animation, we brought up Carbon Cycle 2.0. This is an initiative that was launched by Lawrence Berkeley National Laboratory Director Paul Elvisatos and is now led by Associate Laboratory Director Don DiPaolo. And you can see here some of the examples under the umbrella of Carbon Cycle 2.0, from the highest tech approaches to some that are relying on available technologies. Lawrence Berkeley has had a legacy of smart building, construction, installation, lighting systems, and such. Ashok Gadgil has developed the Darfur stove, which helps those living in Africa derive their energy from the scarce resources there with a very simple stove that's saved many lives. Water purification systems, biofuels that we talked about back in January, and some of the strategies to capture and sequester carbon. And we're going to learn a little bit more about how microbes deal with carbon within the context of the Carbon 2.0 initiative. So let me first take a moment to introduce our panelists. First, we have Jonathan Eisen. That's Jonathan. Jonathan has a biology degree from Harvard, and from there he went across country to Stanford, where he got his PhD in DNA repair. And the DNA repair mechanism is what enables us to be here tonight and not succumb to the various mutations that we'd be tortured by without the code in our cells to respond favorably to mutations. And then Jonathan went back to his native Maryland, I believe, and worked for TIGER, which stands for the Institute for Genomic Research. And he worked with this guy right here, J. Craig Venter, who was also was waving the private industry banner for the Human Genome Project. And as you can see, Jonathan's wearing his beloved Red Sox garb there on the right. And from TIGER, he has moved back to California, where he's on faculty at the Genome Center up at UC Davis, and also has an adjunct appointment here at JGI in Walnut Creek. So Jonathan's research focuses on novelty, and that is not to say novelties, but rather how processes evolve in microbes, and he's going to talk a little bit about that today. He is also an outspoken advocate for open access to scientific literature and one of the editors and founders of the Public Library of Science Biology, PLOS. And he is also, I would characterize him as the Lady Gaga of social networking for science. So he's got a blog and he's got a Twitter feed, and a considerable amount of people follow science from Jonathan. And if he only gained about 9.85 million followers, he'd be up there with Lady Gaga. So our next speaker is Rachel McElprang. Rachel has been on a southerly trajectory since her birth in Spokane, Washington. She did her undergraduate at Utah in biology, got a PhD in genetics from University of Washington, and has been for years now a postdoctoral fellow for our director, Eddie Rubin, his laboratory, and she's studied all sorts of microbes, from those that are contaminating Neanderthal samples to very briefly in the Gulf of Mexico. And then you're going to learn a bit about Arctic permafrost and the implications that microbes have and their role there. And recently she accepted a faculty position at Cal State Northridge, so we only have her for a little bit while longer. And finally, we have Terry Hazen. Terry Hazen, since April 20th, 2010, is probably the microbial ecology equivalent of Lady Gaga, in the sense that he's probably had more interviews on television than he can try to number right now, and we're grateful for him to join us tonight. He has a bachelor's and a master's from Michigan State University in biology, and then went off to Wake Forest for a PhD in microbial ecology. He was chairman of the department, and for 8 years served on the faculty down at the University of Puerto Rico. And he's one of the 4 people who've been honored with the Department of Energy Biological and Environmental Research Distinguished Scientist label. And again, we're happy to have him here. He's got many appointments between Lawrence Berkeley National Labs Earth Sciences Division, where he heads up the Center for Environmental Biotechnology. He's the head of the Ecology Department. He's connected with the Energy Biosciences Institute, whose work you're going to hear about today, and the Joint Bioenergy Institute. He's a man of many institutes. He's also a fellow of the American Academy of Microbiology and has authored over 240 publications and has trained nearly 24 students and 18 postdoctoral fellows. So piloting the discussion this evening is, and I should say, the patents that he has on bioremediation processes that are mitigated by microbes are part of the things he's going to be talking about tonight very briefly. So the pilot for tonight's discussion is John Fowler. John is no science slouch himself. He has a degree in, from University of California, Santa Barbara, and the Air Force Academy. He's done graduate work in experimental psychology and physics, and he's a pilot, and most recently been soaring. He's got 3,000 hours of flight time, and you know him best as in his 35 years of broadcast journalism as the health and science editor for KTVU, and we're delighted to have him here tonight. And I'm going to turn the controls over to John, and he's going to set the ground rules for the night and go from there.
All right, thank you very much, David. My job here tonight is really to get out of the way. I'm going to be your eyes and ears up here, so if these guys get too technical or go too fast, I'm going to raise my hand and try to interrupt. I've already prepped them for it. They very graciously said that it's okay for me to interrupt them. So. The other thing that I want to do is help give you some idea of why this is important. When the current president was sworn in, he said shortly thereafter that science was going to drive policy in his administration. These are three important people providing that science to the lawmakers. Right now, today, we are having a battle royal, if you would, on Capitol Hill between the Senate and the House about the EPA, whether it should step in and is actually protecting the environment, something that they should do with respect to greenhouse gases, for instance, carbon dioxide among them. And so this, what we're talking about, what they're going to talk about tonight, is really crucial. It's timely, it's now, it's very important for all of us to pay attention to this and stay abreast of the science that, in fact, you are paying for. So also, at the end of this, we'll be talking with you. I'm going to ask questions if you have questions. I'll have some, I know. But I'd appreciate it if you'd wait till the microphone gets to you before you holler out, and I'll try to be as quick as I can finding folks to talk to. But really, without further ado, I want to get started with tonight's presentation because it's pretty interesting. I get to talk to smart people all the time. This is really cool. So first, Jonathan Eisen, take it away. Tell us why we're here.
Well, I'm going to have to move to stand up or something because I can't see anything from over there. And he may stop me, but you guys are welcome to stop me too. I'll probably listen to you more than him. But that's, you know, that's just the way it goes. Well, is there a pointer on here too? There is. All right. So what I'm going to tell you about is sort of give you a background and introduction to microbes and the diversity of microbes and some of the things we know about them and some of the ways that we study them as a sort of setup for the more detailed discussions about particular microbial systems that you'll hear about in a little bit. So you may be familiar with microbes through the cushy toys that they sell at some of the stores around here, these giant microbes. If you're really good, I have a whole bag of them over here. To some of the younger members of the audience, we might be negotiating a transfer of them at some point. But they're really cool. And you know, unlike toy lions, and I have two little kids who are obsessed with big cats, all the forms of those, they give you miniature ones, right? A little toy lion and everything. For microbes, we have to come out with these giant ones, of course, because the whole point is that we can't see them. They're small. They don't look like that. Well, some of them do, but most of them don't. And I didn't know there were going to be all these Lady Gaga references, or I would have put a figure of her up there, too. So obviously, as I assume everybody here knows, the point about microbes and the reason that they're hard to work on, in part, is because they're very small. And so when you're walking around in a forest and you see the trees and the squirrels and the other animals, and you can learn about them, and people have learned about the large organisms over thousands and thousands of years, to classify them and understand what they're doing, microbes, we don't generally see them when we're walking around. And they weren't really well understood until people started to look at samples with microscopes. And so this individual in particular wrote a bunch of treatises on this, Antony van Leeuwenhoek. This is one of his little microscopes that he used to look at them. And these are some of his drawings of these microbes that he saw in various samples. He liked to look at the scrapings from his teeth. First sort of human microbiologist, I guess. And he looked at pond water samples and all sorts of other things. And you know, without a microscope, we'd be lost in terms of studying microbes because they're invisible to the naked eye. And so this has created an enormous challenge for understanding what they're doing in the world. Again, because you can go watch a particular type of bird do something in the forest and figure out something about what it's doing, and it's much harder to generally do that with microbes. And now I have broken the system. All right. So when people take out a microscope and look in various places and look at what they see, van Leeuwenhoek saw a lot of diversity, but there's been an incredible amount of diversity that has been discovered over the last 200 or 300 years. He was doing stuff in the early 1700s, and people have been looking at samples from everywhere and looking at what they see under the microscope. And you see all sorts of diverse forms. This is a spiral-shaped bacterium. This is a microbe that secretes a hard shell on the outside and forms these amazingly beautiful structures. This is a multicellular bacterium. So you might think of bacteria, if you have some knowledge of them, as living on their own, but some of them actually form these multicellular structures. There are square-shaped microorganisms, a really bizarre salt-loving organism that people still don't understand how it's able to form these square shapes. There's weird organisms that look like Mickey Mouse on their side. This is actually two species that live next to each other. This is amoeba, the classic sort of thing that was the basis for the blob and other nasty organisms. This is one of my favorite microbes right now because if you search for the name of this microorganism, it's not really a microbe, but it's related to some microbes. If you search for the name of this, which is Xenophytophora, I assume you wouldn't know that in advance, you come up with an image on Wikipedia. This is the picture on Wikipedia that comes up. I have this individual thing in the freezer. It was collected in the deep sea by the Alvin submarine by a graduate student who recognized that this, even though it's almost the size of your fist, is a single cell. A really bizarre organism in the bottom of the ocean. And so right now, actually, I have a graduate student who actually works at JGI, Jenna Morgan, who's grinding this up, I think, a few days ago to try and extract its DNA and sequence its genome. This is a really cool thing called a slime mold that also forms multicellular structures. Just trying to give you a tour of some of the diversity. Even though, again, we refer to them as microbes, this is a single cell that's enormous, so technically it's not invisible to the naked eye. This is actually a bacteria that's about the size of the head of this fruit fly, an enormous bacterial cell. So there is diversity in the size of some of these organisms. We sort of are loose with the definition. We still kind of call them microbes, even though we can see them. It's just easier to not deal with the exceptions in biology in a lot of ways. So anyway, there's this incredible diversity of form among microorganisms. There's also an enormous diversity of kinds of microorganisms. So instead of just the structures of organisms, if we try and classify the microbes into their different types, we can build an evolutionary tree that shows the relationships among all the organisms on the planet. And when people have done this, they've found three main forms of organisms. And they've given them names. So the ones that you're used to hearing about as sort of pathogens that we'll come back to, the bacteria, these are mostly very small organisms that don't have the DNA packaged into a little membrane called a nucleus. The eukaryotes, which includes us, have their DNA packaged into this little membrane and their own lineage. And this third group was discovered in the 1970s by people looking at DNA sequences of microorganisms. And they hadn't recognized that this was a distinct branch on the tree of life until they looked at DNA sequences of different organisms, compared them to each other, and discovered that there was this third form of microbe now called the archaea. And you'll hear a little bit about some of these different groups of organisms, so I wanted to just give you a little bit of a tour of the diversity. Now, there's sort of a slippery slope here. Some people use the term microbe to refer to small organisms that come in cells, as well as viruses. Viruses are parasites that actually don't come in the form of a cell. They have sort of a protein sugar coating on the outside, but they're different structurally from the cells of us and bacteria and these weird archaea. Some people use microbe to refer to all small things. Some people don't like to include viruses in the definition because they're not technically alive. So there's some debate there, but you'll probably hear, I don't know how they sit on the fence of this definition of microbe here, but viruses are really interesting, really important in the environment, and many microbiologists study them as well as these other types of organisms. So now, I'm interested in this sort of diversity of different kinds of microbes, mostly because it helps us interpret the biology that we're studying of microbes. But most of the people that out there are studying microbes are much more interested in the diversity of function that they have. That is, what they're doing. Who cares about sort of whether or not there's an archaea or a bacteria or a eukaryote? We care about what their functions are in particular environments. And so what I'm going to do for the next few minutes is give you just a little tour of some of the different functions that we know about in microorganisms. So, of course, most familiar to most people here are the nasty microbes. And as someone who spends most of their time working on non-nasty microbes, they have all the best names. It's not fair. So, you know, this is bubonic plague, right? I mean, there's not one environmental organism that has a good name like that. This is anthrax, Ebola virus, well, the bacteria that causes syphilis here. So they either have a really cool name or they cause a disease that makes people want to pay for research on those organisms in some way. And so, but of course, microbes do lots of nasty things. They're responsible for disease in plants and animals, including people, and thus there's an enormous amount of research on the pathogenic microorganisms that are out there for good reason. They do all sorts of nasty things. Of course, many of us are also interested in what microbes can do to help other organisms. So in this slide, I have a few examples of systems where microbes are what are called mutualistic. They work with some larger organism and they help each other. So in all leguminous plants, so beans and all of their relatives, these plants are able to grow in nitrogen-poor soil because they have bacteria that go into their roots and fix nitrogen from the air for the plants. And that's what allows legumes to grow in nitrogen-poor soil. And it's actually why people, of course, use legumes to enrich the nitrogen in a particular plot when they're rotating crops. These microbes that live inside their roots are what is carrying out the function. And in general, plants and animals and other macroorganisms, they're kind of incompetent metabolically, biochemically. They can't really do a lot of functions on their own. They have all sorts of interesting structures and behaviors, but they're not very good at carrying out biochemical reactions. And usually when they need a new biochemical reaction, they engage with some type of microbe to carry out that reaction, because the diversity of biochemistry in microorganisms is enormous. Microbes can basically do any type of chemistry you can think of. There's a microbe out there that can manipulate carbon, nitrogen, oxygen, sulfur in some interesting way. So fixing carbon, as this is a session about carbon, microbes help all sorts of organisms pull carbon out of the atmosphere. All plants depend on chloroplasts inside of them, which are used to be free-living cyanobacteria and now have been captured inside plants and algae. That's what fixes the carbon for plants. There's all sorts of other weird organisms like these in the bottom of the ocean that have bacteria living inside of them that fix carbon for these worms. So these giant tube worms in the bottom of the ocean have no mouth, no digestive system whatsoever. They basically function like plants with bacteria that are making all the sugars for them living inside of them. The JGI is very interested in some of the microbes that digest various wood or wood-like products. And in these ecosystems, like inside the cow rumen, which you can fortunately or unfortunately sample through one of these fistulas, at UC Davis, we have a bunch of them roaming around on the campus.
That's a hole.
That's a hole into the side, sort of like when people have ileostomies or something like that, that you can literally stick your entire fist into and collect samples from inside of the cow. And it's what the JGI used in a recent study of the microbes that live inside the cow rumen. Oops. So cows are not actually able to digest the grass that they eat themselves. They have microorganisms that live in their gut that carry out the degradation processes that allow them to live off of this complex carbohydrate that they're eating. And the same is true for termites. And many other wood-eating organisms all depend on their microbes. There's some other really cool nutritional sort of symbioses that are out there. If you grow wine or grow grapes and make wine or live out in Napa, you might have even seen a picture of this. This is the glassy-winged sharpshooter, a sap-feeding insect that transmits Pierce's disease in crops. Inside of its gut, it has bacteria that allow it to feed on nutrient-poor xylem circulatory system, which most animals couldn't feed on. The bacteria make all the nutrients that are missing from its diet for it. So there's a diversity of microbes out there that are mutualistic symbionts for their host that do biochemistry that the plants and animals are unable to do themselves. There's some recently a big upsurge in research on the cloud of microbes, as David already referred to, that live in and on different organisms. So on people, there are more microbial cells than there are human cells. And only in the last, say, 5 to 10 years have people been actively trying to study all the diverse possible functions that are present in these microbes that live in and on us. There's something called the Human Microbiome Project that NIH has been funding to try and characterize the roles that these microbes play in development of the immune system, in digestion and health and resistance to pathogens, in all sorts of diverse activities, using some of the methods that you'll hear about today, but we're not going to talk about the human ecosystem today. Many of the microbes that I work on are the ones that you could call weird, I guess, or unusual in some way. That's sort of consistent theme in my life, I guess. So there are a lot of microbes out there that we call extremophiles because they not only live in extreme environments, they actually thrive in these extreme environments. They can only survive in those extreme environments. And this includes boiling acid pools, Yellowstone hot springs, high salt environments, low temperature environments, low pH environments like Mono Lake, and high temperature bottom of the ocean environments. And in many of these ecosystems, there are microbes in those systems that are doing very interesting manipulations of carbon compounds that were not well appreciated until people started to try and study the organisms in these ecosystems. So in some of the hot springs in Yellowstone National Park and related places, there are microbes that live on carbon monoxide and produce hydrogen gas as a byproduct of their metabolism. Incredibly unusual. Now, in retrospect, we sort of realize that wherever there's energy and carbon compounds and liquid water, there's probably going to be a microbe that's doing something with that energy and that carbon compound and surviving. They may not be surviving great in some of these systems, but in the bottom of the ocean, these organisms can survive at higher than 100 degrees Celsius because the pressure keeps water from boiling. So the temperature gets above 100 degrees Celsius there because of the pressure from all the water on top. Of course, there's all sorts of agriculturally and industrially important microbes out there. Some of the favorites probably for myself and many of the people here include the yeasts that are used to make beer and bread and wine. There are all sorts of different yeasts and bacteria that are involved in making cheeses. And of course, it's not just making food or fermenting various things for us, but we can now take microbes and use them to make or manipulate carbon compounds in various ways. This is one of the avenues of research in biofuel production. So you can feed microbes sugars and have them make all sorts of interesting compounds. And that is because of their diversity of biochemistry, basically. Or it's very manipulable in some way. So of course, the point of this sort of symposium is that in addition to all these other things that microbes do, they kind of run the planet in terms of the global nutrient cycles. They play important roles in all of the important nutrient cycles on the planet, including nitrogen and sulfur and phosphorus and, of course, carbon. So in terms of photosynthesis, for example, the fixing of carbon using the energy of light, that's all basically done by microbes. There are these big leafy organisms that are out there that are incompetent in doing this themselves without the chloroplasts that are inside of them that are basically enslaved or working with microbes. So microbes basically carry out all the photosynthesis on the planet, either directly or indirectly, and much of the carbon cycles on the planet. And you'll hear more about some of their roles in a little bit. So what I just want to take you through briefly here is a little bit about, so I've told you about all this diversity, but I haven't really told you about how people have studied this diversity, which turns out to be both very interesting and very important for what JGI has been doing in the last few years, as you will learn about in a minute. So in terms of microbes out there in the world, you can really oversimplify sort of the field of environmental microbiology and say that people are trying to figure out who is out there. That is, what are the kinds of microbes in different places, just like a birder would try and figure out what are the kinds of birds in a particular place. And what are they doing? What are their functions in particular ecosystems? And it turns out that answering these questions has been much harder than people probably appreciated for many years for microbes. So as a recovering bird ecologist, actually, and butterfly ecologist, I used to go out into the field and do all sorts of studies of various organisms. And this turns out to be both interesting and not, I mean, it's hard, but it's not impossible in various organisms. This is Vladimir Nabokov, the writer who actually was the curator of Lepidoptery at Harvard's Museum of Comparative Zoology, on top of writing novels and other things. You can go out and collect organisms. You can go do studies at a particular site. You can use satellite imagery to learn about plants and plant diversity. You can make nice grids and study the diversity in each of these grids by counting organisms. You can study birds or miss the big picture for studying the little picture. With binoculars, so people actually do butterfly observations now with binoculars in addition to bird observations. And of course, you can do probably more fun things like going into coral reefs and studying them. And you can study plants and animals because you can see them by going out there in the field. Now, it turns out that many environmental microbiologists have figured out ways to study microbes in the field sort of like this. So you can go collect samples in a boiling, I think that's in Yellowstone National Park, sort of a sulfur spring. You can sail around the ocean like Craig Venter did and collect samples along the way. You can use satellite imagery to image some of the products or parts of microorganisms. You can collect soil samples in one of those grid-like patterns. It's not always fun to carry your microscope out to the field, but you can do it and do field studies. And of course, you can use, rather than scuba diving, you can use, say, the Alvin submarine to collect samples in the bottom of the ocean. Now, it's really nice, but it turns out it doesn't work that well. And there are many reasons why it doesn't work that well, but one of them turns out to be really vexing, and that is that the appearance that you would use to try and study microbes in the field is not a very good indicator of the kind of microbe that you're looking at. Whereas with a bird, I have a field guide to birds. I actually left them in my car. I was going to show you all my ridiculous field guides that I have a collection of. You can identify birds very well, almost down to the species level in almost every case, by just looking at them with binoculars. For microorganisms, this doesn't work very well, in part because distantly related microbes can look identical in a microscope. And that means that if you go to an environment and you observe the squiggly thing doing something, and then you go to another environment and you observe a squiggly thing doing something, you don't know how to make sense of it, because you haven't been able to sort of classify the organisms in any obvious way. So this is a big challenge in a lot of environmental microbiology, and there are a variety of other challenges. One thing that people have done instead, or on top of studying microbes in the environment, is to bring them into the lab and grow them. That's called culturing. So you can try and grow microbes in a test tube or on a plate and do all sorts of interesting things now that you have them in isolation from other things in the laboratory. And this is very important to the field of microbiology and turns out to be very powerful, and is what sort of JGI did for many years, sequencing the genomes of microbes that people could grow in the laboratory, and very important to the study of microbiology. But it's also limited in its power for understanding microbes in the environment. And the main reason for this is that it turns out that most of the microbes in any ecosystem, we have no idea how to grow them in the laboratory. So you can go to the environment, you can see things out there, and then bring the sample back to the laboratory, and they don't grow. So kind of screwed. It's very hard to make sense out of a community when you can only grow 1 out of every 10,000 things that is from that community. And to make a long story short, what has developed over many years is indirect means to study the microbes in the environment. And one of those indirect means to study the microbes in the environment has been to collect samples, take them back to the laboratory, isolate DNA from those samples, and use the same methods that people have been using to study the human genome or organisms in the laboratory to indirectly study the microbes that are in a particular environment by sequencing their DNA and comparing that DNA to DNA that we know more about, say, organisms that we've studied in the lab. And this is generally now sort of called metagenomics, the environmental study of genomes of organisms. And this is now being done in all sorts of different ecosystems to try and tackle this massive diversity of microbes. What I didn't really emphasize was the sheer number of microbes that are out there. There are more microbial cells than there are stars in the universe. There's more mass in microbes than there are mass of plants and animals on the planet. And the evolutionary diversity, the number of kinds of microbes, people estimate there are hundreds of millions of species of microbes out there. And we've grown about 8,000 different kinds in the lab. Not a great sample. So there are these sort of systematic efforts by a lot of different institutions to go out and study the microbes in the environment, largely by these DNA or related molecular methods, such as studying the proteins or the RNA in the environmental samples. I coordinated a sort of related project at the JGI called the Genomic Encyclopedia of Bacteria and Archaea. There's this human microbiome project. There's something that's sort of starting right now called the Earth Microbiome Project. It's not just the DOE that's been doing this. The Moore Foundation, the NIH, the Sloan Foundation, all sorts of organizations are supporting DNA-based studies of microbes in the environment because of the potential power to learn about those microbes. And what you're going to hear about is, in part, how people are using some of these technologies, sort of the diversity of technologies that are available to go out and study microbes in the environment, including DNA-based studies, which JGI has pioneered in these environmental samples. So as I said, I'm an ex-birder. I literally have like 6 copies of this book. It's the best bird field guide, by the way. I have one in my car that I keep there all the time, one in my JGI. No, and I may not be in my JGI office anymore. But I have these bird books everywhere. And what a field guide is basically a summary of the types of organisms that are out there, the functions that they have, and their distribution over space and time. It's a field guide to the organisms. What I dream of is that we will get a field guide to the microbes at some point. It's a very hard problem, as you will hear about in part, because of the diversity that's out there and because of the difficulties in studying them. But that's what you're going to hear about now. And again, microbial diversity is amazing, and we're just beginning to get sort of a good handle on particular ecosystems. So I will stop there and hand it over to...
We'll take questions at the end, so please, if you have ideas, write them down or think of...
Or send them to me on Twitter. No.
That'll do it. Go ahead, next up.
Next up is Rachel McElprine.
All right. First of all, thank you very much to the powers that be for inviting me to come and speak. I'm really excited about doing this. I'm excited about the research. And I'm always very enthusiastic about talking about this type of research to the public because I think it's incredibly important to have a well-informed group of voters. So I'm going to talk to you today about the permafrost and the role of microorganisms in the permafrost and in global warming. So first of all, a quick introduction to the greenhouse effect. Essentially, the Earth has a natural atmosphere or greenhouse, and this atmosphere is composed of a number of things, including greenhouse gases. And those greenhouse gases traps the heat that is sent to the Earth by the sun. And this is a good thing. This keeps the planet warm. It keeps the planet habitable. But over the past several hundred years, humans have been increasingly releasing more greenhouse gases into the atmosphere. And people generally think of those greenhouse gases as being released from the burning of fossil fuels. So we are putting essentially carbon dioxide and other greenhouse gases into the atmosphere that weren't there before. And what we're doing is we are increasing the efficiency of our atmosphere. So essentially allowing the atmosphere to trap more heat and causing us to have a warmer Earth. Now, one source of greenhouse gases that people usually don't think about is the permafrost. And the permafrost is poised to become a major source of greenhouse gas release in the next little while. So why is that? Well, what I'm showing you here is a map of the Earth and the amount in the next 100 years that the Earth is expected to warm. And what you can see is that temperatures are predicted to increase dramatically in the Arctic in the next 100 years, much more so than many other regions of the globe.
And just to be clear, that's where the permafrost is.
That's where the permafrost is. The permafrost is defined as permanently frozen ground. So we already are seeing evidence of a thawing Arctic. So what you're seeing here are 2 satellite photos taken of the polarized sheet. One taken in September of 1979, and another one taken in September of 2007. And what you can see is a dramatic reduction in just the short amount of time of the size of the polar ice sheet. We're also seeing other evidence of the thawing Arctic. What you're seeing here are two consequences of the permafrost thawing. So this ground that has been permanently frozen for hundreds and thousands of years is thawing, and it's creating unstable soils. And this is compromising the foundations of buildings that are built on the permafrost, and also creating this phenomenon called drunken forests, where the trees are, that used to be vertically oriented are displaced and are falling over. So why does this matter? Well, it matters because there is a huge abundance of carbon stored in the permafrost. Now what you're seeing here is a map of carbon storage in terrestrial ecosystems. And the darker the brown, the more carbon is stored. And you can see again in these Arctic regions where the permafrost is that there are really incredibly large amounts of carbon stored there. And much of this carbon is in the form of undecomposed plant material. And so it's much like having something in your freezer. You put it in your freezer, and it is no longer susceptible to the microbial degradation processes that it would be if you left it out on your counter. So what happens is that the temperature rises, global warming is occurring, the permafrost thaws, and all of a sudden you have all of this carbon that has been stored and trapped in the soil for thousands of years, that becomes susceptible to microbial degradation. And the byproduct of microbial degradation is the release of potent greenhouse gases, including carbon dioxide and methane, into the atmosphere. And this creates a positive feedback loop to global warming. So the temperature rises, the permafrost thaws, greenhouse gases are released, even more heat absorbs, the temperature rises, and so on and so forth. And so this goes beyond just releasing carbon into the atmosphere. This amplifies the effect of global warming through a positive feedback loop. However, the impacts of thaw are largely unknown. We really have very little concept, first of all, of how much carbon is going to be released into the atmosphere from the thawing permafrost, and at what rate this is going to occur. And this is because the permafrost remains largely unstudied. And the reason the permafrost remains largely unstudied is because these microbial communities that live in the permafrost are extremely diverse. As Jonathan said, we can't grow most of them in lab. We can't take them into laboratory and study them in isolation. And then also the enormous diversity of microbial life in the permafrost creates a barrier. So in 1 gram of soil, you can have upwards of 10,000 species and anywhere from 10 million to 1 billion individual cells. So it's a hugely complex problem that makes this a really difficult and area that isn't well understood. So the question is, how can we study the permafrost microbes? And of course, we're the Joint Genome Institute, and we want to sequence their DNA. So traditionally, how this has been done is to isolate DNA from a single organism and get the DNA sequence. And this is called genomics. However, because we can't grow them, most of these in lab, and because they're so amazingly diverse, we use a strategy called metagenomics that Jonathan touched on a little bit. And what that means is we essentially go out into the environment, in my case in the permafrost, and you take a sample of the soil, and you extract all of the DNA from that soil, regardless of what organism that it comes from. And then you sequence it, and you try to computationally put all that information back together to get an idea of what the functions of the community are. So it would be a little bit like taking a copy of the New York Times and tearing it to bits, and then trying to put it back together again to reconstruct what the stories in the paper were. So through the JGI and the Lawrence Berkeley National Lab, we have a number of ongoing studies to address this question. And I just want to mention here that these ongoing studies at LBL, or Lawrence Berkeley Lab, and the JGI are done in collaboration with Janet Jansen, who's also from the Earth Sciences Division with Terry Hazen, and Mark Waldrop at the U.S. Geological Survey. So what we do is we go up to, we go up to Alaska, and we collect permafrost cores. You can see here is a picture of drilling a core, and here's the core that is taken out. And we can do a number of experiments and studies on those. And we have a couple of goals. And first of all, those are to identify the community members, to identify the individual species that make up the communities in the permafrost, and then to use genetics to figure out what the component parts are. That is, what are the genes or the cogs that do these functional processes that we're interested in. And so eventually, our goals are first to understand the processes involved in greenhouse gas release. That's the very first, because we don't know much about it. The second is we want to be able to predict the impact of permafrost thaw on climate change. And finally, we want to think about mitigation strategies. So possibly biological, and also strategies having to do with public policy. All right, and on to Terry Hazen.
Thank you. Okay, so the Deepwater Horizon. You know, it was on April 20th, the Deepwater Horizon, which was the name of the drilling rig, exploded for unknown reasons and sank, killing 11 people. It was the worst disaster in the oil industry in the United States. There's only a couple that are worse than that in the world even. The floating rig burned for 2 days and then sank. There were 3 breaks in the riser pipe, which is that pipe that comes up from the bottom of the ocean to the drilling rig. Originally placed at 5,000 barrels, and a barrel is 42 gallons. Recent estimates place the oil intrusion into the Gulf at more than 60,000 barrels per day. There was 54.1 million cubic feet of natural gas being flared at the same time. Total release was about 4.9 million barrels. The spill has surpassed the Exxon Valdez spill, which was the largest spill in the United States in Alaska. And it surpassed the Ixtoc 1 blowout, which was in Mexico, in the Gulf of Mexico. And that was 3.3 million barrels. That was the second largest in the world, marine spill in the world. The Deepwater Horizon is now the second largest marine spill in the world, the first being the Gulf War. The well was capped beginning July 12th. With no further input into the Gulf of Mexico after July 15th. And the well was declared dead. Now you will hear me speak about the Deepwater Horizon, the Mississippi Canyon 252, which was the name of the parcel that the well was drilled in. And you'll also hear me speak about Macondo Well. That was the name of the well that was an exploratory well that was actually drilled. All 3 of those things are virtually the same thing. But the drilling rig was the Deepwater Horizon, and that's what most of the media refers to the spill as. Now, if you go to this website, you can see this video work. We tried like mad to get it to work tonight, and I don't know what was going wrong. You can also go to YouTube, and you can see 4 other very detailed lectures, and right in the front you'll see this, and it works perfectly, okay? Basically, it shows here that this is where the Deepwater Horizon rig was, and it's over 50 miles offshore. This is New Orleans proper, and the Mississippi River flowing out here. The rig sank in about 5,000 feet of water, 1,500 meters. And then subsequently, they tried a number of different things, trying to cap it. Virtually none of those things worked until they finally cut off the riser and pipe and then fitted it with a distinctive cap that then caused it to stop leaking oil. The relief wells were drilled, 2 relief wells were drilled starting right away in May, and those were completed in September, and that's when the well was finally declared dead. So what we know happened is that this is really a leak. It's not an oil spill per se like we usually have with a tanker spill, like the Exxon Valdez. It's a very light oil, and it was being jetted into the deep sea where it formed droplets. So one of the things that, I'll show some of this to pass around that you can all see. The oil is a very light oil. So you can see it's sort of a brownish. This is source oil. This is more valuable than platinum right now because that well's dead, okay? And this is the actual source oil. Now, once it's weathered, and you can see it doesn't coat the vessel very well. It's very light. It also has a lot of volatile components and degrades very easily. It's very easy to biodegrade. Weathered oil, it looks like motor oil. And this is what happens once it gets to shore. So by the time it goes 50 miles, it goes up a whole mile from the deep, then goes to shore 50 miles. A lot of the volatile components are gone. A lot of it's been biodegraded. It's been exposed to sun and air and that sort of thing. And so it's what we call very weathered. And this is what we see going to shore, and it becomes emulsified and actually forms a red mousse. And then what happened for the first time ever, we documented that there was a deep sea plume. And this deep sea plume was caused by the fact that droplets that were 10 to 60 microns were tended to be neutrally buoyant. So they were carried with the current at this depth of 900 meters to 1,500 meters, where any larger droplets went to the surface. There was Corexit 9500, or the dispersant, injected initially in the riser, and then finally once they cut off the riser at the wellhead. They also injected a bunch of drilling mud with the top kill experiment, trying to get it stopped, which didn't work, and a lot of junk, and that combined with the oil and is sedimented out around, and we'll talk about that a little bit more later. Of course, there's a lot of natural seeps, and we'll talk about that more later. There was a well operation command. They were skimming, they were burning, they were adding dispersant. There was natural weathering going on, and so we had all of these things going on at the same time. Also, this is a paper that was just recently published. This shows how much natural gas was part of this. Over 40% of the mass was actually natural gas or methane, and you can see how much of that, of course, being dissolved and going into liquids and then going to the surface of that oil, and the gas being flared from the recovery well, and then forming CO2, carbon dioxide, carbon monoxide, carbon black, and of course these alkanes and aromatics could also be released, and of course some of it went to shore also. So tremendous operation. This was the biggest response and remediation operation for any oil spill ever. There is no doubt in anybody's mind. 4.9 million barrels, 31,800 people involved, 3,447 kilometers of containment boom. They cornered the market on containment boom worldwide. You could not find any anywhere at one time last summer. 441 controlled burns and 1.84 million gallons of dispersant used. And I'll talk more about that later. What we did is to use our systems biology approach, looking at the hydrocarbons and their stable isotope and nutrients, physical chemical analysis. We also looked at the fats. We looked at, if you remember your basic biology, we had DNA forms RNA, forms proteins, and so on, and lipids, and we looked at all of that. So we looked at the DNA, we looked at the RNA, we looked at, did single cell isolation with JGI, did a lot of sequencing, cloning, and phylochip, and a variety of these technologies. The important thing was here, we had to use multiple lines of evidence to prove exactly what was going on there and to prove that there were organisms down there that could degrade that oil in that deep water plume, which we did. I have over 2,000 pictures from the Gulf, from the various ships. We were on 4 different ships. We were down there continuously from May 25th to October 20th, continuously. And Rachel was down there with us. And it's something she will always remember.
I'll never forget it.
You can see the burning that was going on here and the off-gassing when they were capturing some of the oil. They were flaring the methane. They had to have 2 ships constantly spraying water to make sure that they didn't have a problem here. With fires. This is the oil being released at the blowout prevention device, which completely failed. Most of the time what we saw was sheens on the surface like this. But occasionally we did see red mousse. And this is the emulsified oil. We also took sediment samples and then along the shoreline and are looking at that once the oil came to shore. We captured water in CTD, conductivity temperature depth device, which could be lowered in the water column all the way to the bottom. It had several different instruments on it. And then for surface samples, we used the Halliburton bucket, which seemed appropriate somehow, you know. And you can see the oil here, and you can see 2 of our postdocs collecting samples here on the Ocean Veritas. And this is some of the surface oil water samples that we filtered and immediately flash froze on board the ship. Here again, we're taking samples. We're right near the wellhead, and we're taking samples on the CTD. And these bottles can be fired at certain depths and take water samples, and then we can filter them when we come back to the surface. So on 2 different ships, we took a variety of samples, and this paper was published in Science in August, and it came out in August 26th in Science Express. And it was the first time ever that anybody had reported exactly what goes on with these deep water plumes and the fact that there's microbes down there. Basically, microbes can degrade oil. It takes a community to some extent to get complete degradation to carbon dioxide and water. One particular bug may only produce daughter products, or they may take some, break down some of the oil hydrocarbons, and then of course what's left is sort of a daughter product. Other bugs can, and when I say bugs, I mean microbes, other bacteria can then degrade some of these daughter products. So we can get complete degradation if we have proper nutrients, pH, temperature, and oxygen. We used a phylochip, which is an invention that we have at Lawrence Berkeley National Laboratory. And we can immediately detect with this array 50,000 different bacteria and archaea in one test. The beauty of that is we can do it overnight. So we don't have to wait a long time for sequencing. We can see exactly what species are there. We can't see what enzymes are there and that sort of thing. But it's a very good way for us to screen a large number of samples and to see exactly what the concentration of the bugs are without doing anything other than extracting their DNA. We found bugs down there. And as I'll show you in a minute, was no great surprise to me. This was one only 1.3 kilometers from the wellhead, and you can see they're already started to divide right here. And so this bug is about 20 microns long, and it's in complete division. But once we get out to 10 kilometers, they're fat and happy, higher densities, and this is basically the same amount of water that was filtered through this filter. You can actually see in this case the bacteria growing on the oil droplet. This is an oil droplet of Macondo oil. And the resolution on this screen is not very good, but this is in a bunch of publications. If you go to the web, you can find this same picture. You can see this strand of bacteria actually attached to the oil droplet, literally sort of sucking it out and degrading it. What happened to the oil? Well, we showed that the bacteria were down there, that they were degrading it as fast as a half-life of 1.6 to, or 1.2 to 6.1 days, okay? That's how much, how fast they could degrade it. Some people were surprised by that, but we showed in the literature from other Arctic studies and that sort of thing that that was actually similar to other studies that have been reported. That doesn't mean that in 2.4 days, for example, remember I said 1.2 would be the maximum rate. It doesn't mean in 2.4 days all the oil is gone. In 1.2 days, I said half-life. That means half the oil is gone. In 1.2 more days, half of the remaining oil is gone. Because there are some components that will degrade much more slowly. And that's what we're left with some of those daughter products. We went out looking for the oil from July 27th to August 26th. Remember, the well was shut off in July 15th. And we couldn't find it. It was already gone in the deep. And this corresponded to the rates that we saw in the dilution rates. We went out 300 kilometers from the wellhead, did transects trying to find where that doggone oil went. It was gone. It had disappeared. We couldn't detect it. And this is from as little as 1 ppm in our fluorescence detection on board the ship. And then we took samples back to the lab and analyzed them down to 2 parts per billion, 170 samples, and couldn't detect anything. And this is in all the government reports. And by the way, I testified before the Deepwater Horizon Commission, the President's Deepwater Horizon Commission, and it is in all of the government reports too. Why? There is more natural seeps in the Gulf of Mexico than almost anywhere else in the world. 400,000 barrels to 1 million barrels a year go into the Gulf of Mexico every year, okay? Tremendous amount of oil.
Naturally.
Naturally, yeah. And so if I had to look for bacteria that would be adapted to degrade oil, heck, this would have been the place, okay? I mean, it didn't surprise, all the reporters kept saying, doesn't this surprise you? No. I mean, this would have been the place. The other place I might have looked is off Santa Barbara, where if you've ever been down there, you see tar balls on the beach and that sort of thing, because there's natural seeps out there too. But look up here in Alaska, nothing. And that may be part of the reason why the Exxon Valdez spill, things degraded much more slowly, but it was also heavy crude, as we'll talk about in a minute. So, these are images that are going to be hard for us to forget. And this one image has been downloaded from the web over 7 million times. And I don't know if this poor pelican survived. Maybe he did. But all oil spills are local, like all politics. So if we look at some of the major oil spills, including the Casco-Busan spill here in San Francisco, which was minuscule compared to all these other ones, 2,500 birds were killed. 6,000 birds, according to the latest count, were killed in the Deepwater Horizon. But Exxon Valdez, 250,000 birds, 22 killer whales, 2,800 otters, and God knows how many fish, okay? And that was only an order of magnitude, it was an order of magnitude lower than the Deepwater Horizon. So the type of oil going to shore very rapidly, whether dispersants were used, a variety of things like this can affect it. Also, the Ixtoc 1, which was in the Gulf of Mexico, 3.3 million barrels. Again, our best estimate is only about 10,000 birds were killed. That's a lot. These are catastrophes. But as we said quite early on, you know, it's not the gloom and doom of the Gulf. It will recover. And we're seeing that already. And you see the news media has dropped that mantra already. The only thing that we're seeing is close to shore where some of that oil escaped and got into shore. We should have spent more time skimming and keeping that oil from reaching shore. Once it reached shore, sunk into the sediments, it can cause a lot more problems. This just was a recent report from the Texas A&M, and it shows the amount of coastline that was oiled and the effect from Ixtoc 1 down here in the Yucatan. In the red, you can see all the coastline that was affected. People didn't even remember Ixtoc 1. They kept comparing it to Exxon Valdez. And in fact, I'll tell you, I was interviewed by ABC News and Diane Sawyer's producer down here on campus at Berkeley. And I told them early on, I said, once the well is capped, it's going to be more like Ixtoc 1 because it's a light crude. The bugs are adapted. There's a lot of circulation going on here and that sort of thing. It's going to go away pretty fast. And he said, well, how fast? 30 years or 20 years, 10 years, 5 years? And I said, it's going to be less than 5 years. He didn't like that answer. And they told me 7 nights in a row that piece was going to be aired. It never was. Okay, so, you know, there was a lot of gloom and doom in the media. And the fact is, what they should have been comparing it to is Ixtoc 1, which went on for 9 months, was 50 miles offshore. It wasn't as deep, oiled more, but the bugs were adapted. They used a lot of dispersant at the surface, and it was actually similar. Now, there may be some more sensitive areas here in the coast that may be affected by BP, and this will take longer term. It was an ecological disaster. There's no doubt about it, okay? And I don't want to play that down at all. Now, a lot of people ask me about the dispersant. How bad was it? A lot of people said it was very toxic. Okay, so there's a few older people here. So those of you that have had a colonoscopy, okay, the night before, you received something, propylene glycol, which was a really effective dispersant. And I have had that procedure done, and I can tell you it is really effective dispersant, okay? That's a major component in this. It's not toxic. I hope our physicians aren't giving us anything that's toxic, but it is a major component of Corexit 9500. And it's water soluble, and it disperses this material quite well. We couldn't find it. We still can't find it in any of the fish. We can't find it in the sediment. We can't find any sediment samples that are less than, only the ones that are within 1 kilometer can we even find oil down deep. Now, some other people have said that they see some oil farther away. You've probably seen some of the reports of that. Until we see their data, we don't know. I can't resolve those differences. They may have sampled different places at different times. We may have missed something. But we took 1,440 sediment cores and only found contamination significantly within 1 kilometer. And it was all associated with the drilling mud. So the bugs can do some good for us. Would I recommend using any of these bugs for bioaugmentation or synthetic bugs? No, no, no. We showed you how fast they can degrade it. Mother Nature does a pretty good job of cleaning herself up if we can get the neat material up, and that's what we needed to do. But once we get that neat material up, leave her alone and let her do the thing. And that's going to be the hardest thing that's going to be to educate people about, is that sometimes the best thing to do is nothing at all, because nature does take care of itself a lot of times. Thank you.
Before we get away, I have a question. How about taking those bugs that, or microbes, that you find in the bottom of the sea in the Gulf, and transport them to where oil spills occur? Will they live in those places? Can you do that?
So for oil, normally within any oil spill anywhere, within 72 hours we see a major increase in the number of petroleum degrading bacteria. And those bugs are adapted to that particular environment. They're found there naturally, so while there may be some limiting conditions that we may be able to help them out with in terms of iron or phosphorus, phosphate for example, and most of the time we don't need to do anything. Now, what we have found when we take and do bioaugmentation, we grow these bugs up in the laboratory under laboratory conditions. And then we put them out in these places where they're not adapted to those environments. They die immediately. And then they become nutrients for the indigenous bugs. That's an extremely expensive way to make fertilizer, okay? Really expensive. And so that's why you don't see any of that adaptation. And you see us talk about that in this American Academy for Microbiology report that we did, frequently asked questions about microbes and oil spills.
Thank you. Does anyone have any questions? Well, let's start with Terry. And do we have microphones here? Oh, perfect. Thank you very much.
Mr. Fowler, actually, my question is for you.
Yes.
I can't help noticing you've got a pair of black boots on.
Yeah.
Yeah. Texans call crude oil black gold or Texas tea. Can you tell us what Texans call black boots?
I have no idea, frankly. I'm from California.
Well, quite simply, real Texans use black boots out in the ranch to kick around cattle dung and horse dung.
I'll make sure I clean my...
You have a hint of what they might call it.
Okay. Any other questions for the panel? Yes, sir, right there.
So I've heard there's a large dead zone at the mouth of the Mississippi due to the agricultural runoff and all that sort of stuff. To what extent does that affect the ecology of degrading the oil.
Yeah, it does, and actually if you go to the YouTube, you'll see the longer version of this lecture with some more details about the dead zone. The plume down deep was running to the southwest. The dead zones were up to the northwest in much shallower water. There was no effect that we could see at all with that oil moving into the dead zone area.
Except what about near the coast, the stuff that made it to the shore?
No, there wasn't enough. So the amount of oil down deep actually didn't cause any depletion of dissolved oxygen. We saw DO dips, what we call dips, so significant decreases, but they were nowhere near being, anywhere near toxicity or anything for any fish or wildlife.
On your right, John.
I'm sorry, am I right? Over here?
Hi. Two questions. One kind of dumb, which is if the atmosphere is trapping greenhouse gases, is it possible, like with an ozone hole, just to poke a hole in the atmosphere and let the gases out? And the second question is that if the Gulf of Mexico is so friendly to oil-destroying microbes, is it possible to use this science to say, well, where we will drill in the United States is where it's the most microbe-friendly? So in other words, don't drill in Anwar, but drill in the Gulf.
You know, I got asked that question a lot, and I don't, this doesn't give the oil companies a free pass, to drill in the Gulf. They still need to be very careful, and this was definitely a disaster. Now, what we did show, and what Kessler and Valentine showed in the paper in Science in January, is that once the oil was degraded, that because it's such a good carbon and energy source for the bacteria, they'll degrade that first. But then they switched over to the methane because there was a lot of methane down there and there was nothing else, all the oil was gone. And so we saw tremendous biodegradation rates of methane. In fact, enough so that even though there were tremendous releases of methane here, and this occurs in the Gulf, it was all being degraded before it reached the surface. So it was being converted at least to CO2. Now, methane is 20 times more a greenhouse gas than CO2 is, so that could be good news for release of methane in some of these areas that have natural seeps, at least natural methane seeps and that sort of thing. That probably will not be true in the Arctic, from what we know, even in the Arctic Ocean, because there's not enough natural seeps there to maybe adapt those bugs to have a memory for degrading oil.
And poking a hole in the atmosphere almost certainly won't work. The ozone hole simply lets UV in rather than letting things out, for example. I mean, the stuff sort of settles, but it's going to be very hard to make it escape.
Rachel, that what was in your graphic that was that she's speaking about. So we should understand that UV comes in, but infrared is trying to get out.
Right, and that's what the atmosphere traps.
Yes, and so infrared makes heat. That's why it gets warm.
Exactly.
Bugs in the, or microbes, I'll say, in the permafrost, what do you want to learn about this? What do you want to do with this information?
Well, the most obvious thing to do, and the first thing we want to do, is once we get a basic understanding of what they're doing, is use that in climate models. Because current climate models that are out there that forecast just how bad global warming is going to be, and therefore the impacts, say, like of rising sea levels on, our port cities, those types of things, is we really need to be able to predict what the impact of permafrost thaw is going to be, and just how badly that's going to impact global warming. So that's kind of really the first thing that we want to do.
So we want to know what's going to happen.
We want to know what's going to happen. Right. Yeah, and Terry was mentioning methane. One of our studies that we've done, what we showed is that in the permafrost, even under these frozen conditions, that there are microbes that generate methane over long periods of time, and this methane is trapped in the permafrost. And then when the permafrost thaws, the methane starts to be released. But there's a dynamic where there then are other microbes that consume the methane and release CO2 instead. And so it's kind of this community dynamic that goes on.
Wow. Let's go to any questions.
Another question on the right.
Again on the right, yes, ma'am.
Actually, it's a question for Rachel that falls right on what you're saying. Do all of the plant matter you were speaking about that's trapped in the permafrost, is that all that's been built up from times in the past when maybe it was warmer, and so it's like, you know, a history full of plant matter, or is that also or, you know, just things that are growing there but never really decompose?
A little bit of both. So the permafrost accumulation the carbon accumulation in the permafrost can be a record of plants from long times past. And it also can just be because the plants that are growing in those areas, you know, there is a yearly cycle where plants grow. It just doesn't decompose fully. And there are processes like something called cryoturbation, where the ground isn't static, and so it kind of moves, and a lot of that carbon is moved downward during that cryoturbation process.
Are you also thinking that you might be able to, and I mentioned this earlier, tinker with the microbes and try to make them more like Terry's microbes and do good things?
Well, I don't see us engineering any microbes to solve this problem. You know, when people talk about mitigation strategies in terms of limiting the amount of greenhouse gas released from the permafrost, I think a lot of them are talking about public policy. But then I've also heard kind of ideas thrown out there, like one of the reasons that the permafrost thaws is because global warming is increasing the snow cover in a lot of these areas, and this increased snow cover acts as an insulator. Oh. And so
counterintuitive, yeah.
Yeah, it is counterintuitive, but it acts as an insulator, and so people have talked about building in those areas that are susceptible to that, kind of these fences or places that can control the snow cover. And then I've heard people talk about maybe inoculating different areas with, say, microbes that can consume methane, because you'd rather, you know, methane is much more potent than carbon dioxide as a greenhouse gas. And you'd rather have microbes consume the methane and release CO2 than have methane released directly into the atmosphere.
So, Rachel, one of the things that I've heard too is that there's more carbon trapped in the tundra and in the permafrost than in all of the tropical forests combined.
Yes.
Isn't that scary?
Yeah. There's more carbon in the permafrost than there is in the atmosphere.
So if all that goes to methane, we're in big trouble.
Yeah.
My goodness.
I mean, it really, the scale of this problem is potentially huge.
Yeah.
How rapidly is it warming?
Well, it depends on where you look. So there are study sites that we have in Alaska where the permafrost is near 0 degrees, or 0 degrees Celsius, so near the freezing point. And it's warming at maybe 0.1 to 0.2 degrees per year. And so it's just going to be within a handful of years that a lot of this permafrost that was previously frozen is going to be thawed. And in fact, we have a number of study sites where the depth to the permafrost, you know, in all permafrost sites, there is an overlaying active layer that freezes and thaws annually. So the depth of this active layer is increasing, which means that areas that used to be permafrost are now thawing. So it's already happening.
We've got to let you go to get back to the lab.
John, sorry to say we've run out of time, but just 2 comments. One is that I want to thank Walnut Creek TV that has been capturing this evening's event. And if you go to walnutcreektv.org, you can capture the times and places that you can see it. Also, we'll link to it on the JGI website. And I also encourage you, on the way in, you may have gotten a hold of one of our surveys in paper form. But if you're more inclined to use the electronic version, there's both the URL up here and this fancy QR signature here. If you point your your cell phone at it somehow. That'll capture that and tell us all about it. And please be frank and let us know what else you'd like to hear from us. And again, why don't you join me in thanking our panelists tonight for a fantastic show.